Hello everyone, welcome back to the channel. In our last video, we said that in metabolism, we are having two metabolic pathways. One is called as catabolism and other one is called as anabolism. Catabolism is the process of breakdown of a larger carbohydrate molecule to give its simpler components. In case of anabolism, the simpler components will combine together so as to produce the complex carbohydrate molecule. So those things will be observed in case of carbohydrate metabolism. So in this video, we shall give a detailed discussion regarding the carbohydrate metabolism. So we know that carbohydrates are the major energy sources. So whenever the energy is required for a body, the carbohydrate molecule will involve in required biochemical reaction, and that releases energy, and that will be utilized in order to carry out the cellular activities as well as to maintain the body temperature. And the simplest carbohydrate molecule is glucose itself. We know that glucose is having molecular formula C6H12, and this glucose is very rich in potential energy. When this glucose undergoes complete oxidation, in producing carbon dioxide and H2O, that is water, so there occurs releasing of a large amount of free energy, in a strictly speaking way, standard free energy, that we can be given in the form of delta G naught, standard free energy change, and that is of the factor of minus 2840 kilojoules per mole. Mean for 1 mole of glucose molecule, for 1 mole of glucose, there will be release of 2840 kilojoules of free energy. So the negative sign what we are indicating over here accounts for the releasing of energy when glucose undergoes oxidation to produce carbon dioxide and water. And whatever the food we consume, the carbohydrate we consume, that finally break down to give glucose molecule, right? And the cells will store the large quantities of glucose as high molecular weight polymer. Say for example, starch. This will be produced in case of plants. And if you come to the case of animal cells, in animal cells, the glucose content will be stored in the form of a complex polymer, that what we call as glycogen. So glucose will be stored either in the form of starch in case of plants, and in case of animal cells, the glucose will be stored in the form of glycogen. Okay, and when the cells are in need of energy, glucose will be released quickly from this complex polymer molecules, what we can also call them as storage polymers, and the released glucose will undergo oxidation and produces huge quantity of energy, and that will be used for carrying out the cellular activities as well as once again to maintain the body temperature. And here we said glucose will undergo oxidation and produce huge amount of free energy, which is useful for carrying out biological activity. But the work of glucose is not only in producing the free energy. Along with supplying energy, glucose is having capability in supplying a various number of metabolites. In simple language, metabolic intermediates. And those metabolic intermediates that are going to produce by glucose are very much useful in biosynthetic reactions. So that is the specialty of glucose. And if you give a careful observation, glucose is having a total of 3 metabolic pathways importantly. So glucose is having 3 important metabolic pathways. Number 1, the glucose will be stored in the form of polysaccharides. That is, glucose will be stored as polysaccharides. So if you come with the word polysaccharide, the materials that comes to our mind is starch and glycogen itself, or even in some cases you can go with cellulose also, or even in some cases the glucose molecule may changes to produce sucrose also. So this is the first metabolic pathway of glucose, and in second metabolic pathway the glucose molecule will undergo oxidation. So the glucose molecule will undergo oxidation, and through this oxidation it will produce a 3 carbon atom compound which is called as pyruvate. So glucose will undergo oxidation so as to produce pyruvate, and this process of production of pyruvate from glucose through the process of oxidation is simply called as glycolysis. And the third metabolic pathway of glucose is, once again, the glucose will undergo oxidation, but here in this case, through the oxidation, glucose molecule will not produce pyruvate. Instead, it will produce pentose phosphate. Pentose phosphate. So here we said, through oxidation, glucose will produce pyruvate, which is a 3 carbon compound. And here, through oxidation, it will produce pentose phosphate through the pentose phosphate pathway. So these are the 3 important metabolic pathways in respect of glucose. And here in this video, we shall give a detailed discussion about the one important mechanistic pathway, the one important metabolic pathway of glucose, which is nothing but glycolysis. So now we shall proceed further with the term glycolysis. The term glycolysis is actually derived from the Greek word glykis and lysis. So from these two words we have derived the word glycolysis. So the meaning of glykis is sweet, and the meaning of lysis is a well-known word. The meaning of lysis is breakdown. So collectively we can say the meaning of the word glycolysis is simply nothing but breakdown of the sweet. So breakdown of the sweet, we can easily say sweet is nothing but glucose, which is the simplest carbohydrate molecule. So we can simply say glycolysis is the process of breakdown of glucose into smaller component. So here in glycolysis, glucose will splits to give a simpler molecule, and this glycolysis is an almost universal central pathway of glucose catabolism. So glucose is a glucose catabolic pathway. So we have already said in the very beginning of the video the meaning of catabolism. Catabolism is a process of breakdown of a larger carbohydrate molecule to produce simpler molecule. So here in glycolysis, glucose will get breakdown to give simpler molecule. So if you consider the breakdown of the glycolysis, if you go with a peripheral idea of glycolysis, one molecule of glucose in glycolysis, one molecule of glucose will be degraded to give a 3 carbon atom molecule which is called as pyruvate, and this conversion of glucose into a final product, that is pyruvate. So in glycolysis, glucose will be changed to produce pyruvate. So there will be a total of 10 reactions for the conversion of glucose into pyruvate. So these 10 reactions, each and every reaction, each and every degradative stage is catalyzed by specific enzymes. So in a strictly speaking way, we can say glycolysis is a enzymatic catalyzed degradative pathway of glucose in producing pyruvate. Okay, and when in case of glycolysis, during the conversion of glucose into pyruvate, in certain stages there occurs releasing of definite amount of free energy, and the free energy which is released in case of specified steps of glycolysis, that will be conserved in the form of adenosine triphosphate, that what we call as ATP. Okay, so the free energy which will be releasing in the conversion of glucose into pyruvate during the process of glycolysis, that free energy will be stored in the form of ATP. And here we said there are a total of 10 reactions in case of glycolysis, starting from glucose and ending with pyruvate. A detailed mechanistic pathway, detailed reactions of glycolysis was actually clearly explained by three scientists by name Emden, second person is Meyerhoff, and third person is Parnas. So these are the three scientists who gave a very good contribution in explaining the reactions of glycolysis. So by honoring them, the reactive pathway which was given by these three scientists in explaining glycolysis is simply called as EMP pathway. EMP pathway of glycolysis. So this is an universally accepted pathway in explaining the process of conversion of glucose into pyruvate. Glycolysis will occur in both aerobic as well as in anaerobic conditions. And the total amount of energy, that is the total amount of ATP which is going to produce in case of glycolysis, in case of aerobic and anaerobic condition will get differ. And in order to know at which stage, at which conversion the ATP production will occur, and in which stage is there any consumption of ATP, and how much of ATP will produce in a particular step, actually at which stage the ATP production will occur, and what are the enzymes that are going to involve in making a specific conversion. In order to know all these things, we have to go with the reactions of EMP pathway, that is glycolysis. But before going to the detailed reactions of glycolysis, first let me give you a simple flowchart so as to remember which are the sequential conversions that occurs in glycolysis one after the other, and that will be helpful for us in drawing the reactions that actually proposed by EMP pathway in explaining the glycolysis. So now we shall go with the flowchart of glycolysis, then we shall say about the detailed reactive pathway. Now we shall write the flowchart. So first, the glucose molecule will be obtained from the polysaccharide. Now we shall go with animal cells. In animal cells, the polysaccharide material, the glucose will be stored in the form of glycogen, and glycogen will degrade to give glucose. And first, the produced glucose molecule will get activate through phosphorylation so as to produce glucose 6-phosphate. So this is the first reaction of glycolysis. So glucose will get activated through phosphorylation so as to produce glucose 6-phosphate, and this glucose 6-phosphate will get isomerized so as to produce fructose 6-phosphate. This is the second conversion, and this fructose 6-phosphate will again undergo phosphorylation for the second time, and so as to produce fructose 1,6 diphosphate. And fructose 1,6 diphosphate will get degrade, will get split up so as to produce two components. One is glyceraldehyde 3-phosphate, and one more dihydroxy acetone phosphate. Dihydroxy acetone phosphate. Dihydroxy acetone phosphate. So these are the two materials that are going to produce by the splitting or breakdown of fructose 1,6 diphosphate. This way. And this dihydroxyacetone phosphate will get isomerized so as to produce glyceraldehyde 3-phosphate once again. So this glyceraldehyde 3-phosphate will reversibly get changed so as to produce 1,3 diphosphoglycerate. 1,3 diphosphoglycerate. And this 1,3 diphosphoglycerate will lose one phosphate unit so as to produce 3-phosphoglycerate. 3-phosphoglycerate. And this 3-phosphoglycerate will get mutate so as to produce 2-phosphoglycerate. So just the position of the phosphate unit will get change over here from third position to second position, and this 2-phosphoglycerate will once again get changes into the penultimate compound called as phosphoenol pyruvate. And this phosphoenol pyruvate will finally changes into pyruvate. So these are the total conversions. So here you can notice the total of 10 conversions. So 1, 2, 3, 4 and 5. So 5 reactions, 6, 7, 8, 9, 10. So as I told you in the very beginning, there are a total of 10 reactions that occurs in case of glycolysis. But here these 10 reactions have been divided into 2 stages. The first 5 stages, that is from glucose to glyceraldehyde 3-phosphate, inclusive of interconversion of dihydroxyacetone phosphate, which can be simply abbreviated as DHAP, DHA. Okay, so these first 5 reactions are called preparatory phase. It is called preparatory phase. And the next 5 reactions starting from here, glyceraldehyde 3-phosphate to this 1,3 diphosphoglycerate, 1, 2, 3, 4, 5. The remaining 5 reactions, I mean to say the next 5 reactions is called as payoff phase or payoff stage. The reason for calling the first 5 reactions as a preparatory phase and the next 5 reactions as a payoff phase is, in this first stage, in converting glucose into glyceraldehyde 3-phosphate, there occurs consumption of ATP molecule, and in the next 5 reactions, in the next stage, there occurs releasing of ATP molecule, and that's why in the first stage we call the first 5 reactions as a preparatory phase, and the next 5 reactions are collectively called payoff phase. And whatever the conversion here I have shown, that is from glucose to pyruvate, up to here it is common for both aerobic and anaerobic condition. But compared to aerobic condition, in case of anaerobic condition, there occurs one more conversion from pyruvate onwards, that is pyruvate reversibly get changes into lactate. This is in case of anaerobic condition. So this is specifically occurs, this particular stage is specifically occurs in an aerobic condition. This is an extension compared to aerobic pathway of glycolysis. So here you can get a simple idea from what is the first conversion and what is the final reaction, and up to what I am supposed to consider in case of aerobic glycolysis, and what could be the extension of glycolysis in case of anaerobic condition. So if you remember this flowchart for the EMP pathway of glycolysis, then it will be easier for us to write down the detailed reactions for EMP pathway of glycolysis. And do remember, we said each and every reaction of glycolysis is catalyzed by specific enzymes. And now we shall go with the detailed reactions for each and every conversion. Now we shall see the detailed reactions of glycolysis, and in doing conversions of one form to other form, we shall see which are the enzymes that actually take part in making the conversion of one precursor into other. So the first conversion in EMP pathway of glycolysis is conversion of glucose into glucose 6-phosphate. And here we can notice glucose is converting into phosphate, means glucose is undergoing phosphorylation. So here there should occur utilization of ATP in converting glucose into glucose 6-phosphate, and that we shall see in reaction. So here occurs glucose. We have taken the Haworth structure of glucose, and we said glucose has to be converted into glucose 6-phosphate. That means phosphorylation should occur, and in order to phosphorylate a carbohydrate molecule, we need utilization of ATP, that is adenosine triphosphate. And phosphorylation will be always an irreversible process. So glucose phosphorylate to give glucose 6-phosphate. See, the 6th position of the glucose will get phosphorylate. And now we shall write the Haworth structure for glucose 6-phosphate. So here 1, 2, 3, 4, 5, and this is the 6th position, and this 6th position will get phosphorylate, and the phosphate will be written in the form of PO3 2 minus. So here H, OH, H, OH, OH, H, and here it is H, and here it is H and OH. So glucose have been converted into glucose 6-phosphate. So P refers to phosphaturia. And in order to carry out phosphorylation of a material, the enzyme required will be kinase enzyme. And here the material we are using is a hexose sugar. So the needy enzyme in order to phosphorylate glucose into glucose 6-phosphate with the utilization of ATP will be hexokinase, and the type of hexokinase present is glucose. So the enzyme that will be utilized over here is glucokinase with the presence of activator Mg2+. So in presence of glucokinase, glucose will undergo phosphorylation so as to produce glucose 6-phosphate. And here we have utilized ATP molecule. So there will be 3 phosphate units. Among the 3, 1 have been given to the glucose molecule in order to phosphorylate the 6th position. So after that ATP will be producing to ADP. This is the first reaction of glycolysis. And now we shall see the second reaction. The second conversion in case of glycolysis is the glucose 6-phosphate, which is produced in the first stage, first reaction, that will be changed to fructose 6-phosphate. So you can notice glucose and fructose, these two are isomeric form to each other, and the position of phosphorylation is also same. So we can easily predict the enzyme that should be present over here in making conversion of glucose 6-phosphate into fructose 6-phosphate. That means it should be an isomerase enzyme. And now we shall write the reaction for this conversion, and now we shall see what is the actual enzyme that will be utilized in making conversion of glucose 6-phosphate into fructose 6-phosphate. So in second reaction, the glucose 6-phosphate which is produced in first reaction will be reversibly changes into fructose 6-phosphate, and here for fructose 6-phosphate also we shall write the Haworth form. So the Haworth form will be CH2OH, OH, and here H, OH, and H, and at the 6th position there is a phosphorylated carbon CH2O PO3 2 minus, that is fructose 6 phosphate. So here you can notice glucose is converted into fructose. As I said, these two are the isomeric form, and the reactant material, the substrate material is glucose 6 phosphate, that is phosphorylated glucose, and that is converting into fructose. So we know that while giving the names for enzymes, we should notice the type of reaction happening and the nature of the substrate material which is undergoing transformation. So this is a phosphogluco material which is isomerizes to give fructose. So the name of the enzyme material that converts glucose 6 phosphate into fructose 6 phosphate is phosphoglucoisomerase is the name of the enzyme that has been utilized over here in making isomerization of glucose 6 phosphate so as to produce fructose 6 phosphate. Now the third conversion is, so the fructose 6 phosphate which is produced in reaction 2 will be again undergo phosphorylation so as to produce fructose 1,6 diphosphate. So again phosphorylation should occur, and you can notice again the phosphorylation is an irreversible reaction, and again here we need the kinase enzyme. So the name of the enzyme utilized and the structural details of the substrate and product material for the conversion of fructose 6 phosphate into fructose 1,6 diphosphate we shall write now. So here we have fructose 6 phosphate, and this fructose 6 phosphate has to be converted into fructose 1,6 diphosphate. So phosphorylation should occur, and that takes consumption of one more ATP molecule. Once again it is an irreversible reaction. The sixth position is already phosphorylated. Now the first position will get phosphorylate so as to produce fructose 1,6 diphosphate, and we shall write the structure of fructose 1,6 diphosphate, and here the first position is phosphorylated OH, and the sixth position is already phosphorylated, that is CH2O PO3 2 minus. So these are the structures of reactant and product in case of reaction number 3, that is fructose 6 phosphate is converted into fructose 1,6 diphosphate, and here the enzyme required, so fructose 6 phosphate is undergoing phosphorylation. So for the phosphorylation process the enzyme required is kinase, so phospho form of fructose, and the process of type of reaction is phosphorylation. So the name of the enzyme utilized over here is phosphofructokinase. So phosphofructokinase is the enzyme, and again the activator will be Mg2 plus. So again we are noticing the third reaction is once again a process of phosphorylation. So you can notice in the first reaction we have noticed the consumption of ATP molecule, and similarly in the third reaction also we are noticing the consumption of ATP molecule. That what we already said in the very beginning of the video, that is the first 5 reactions are called as a preparatory phase because there involves consumption of ATP molecule in this stage. So now we shall go with the fourth reaction. So what is the fourth reaction? Fourth reaction is the fructose 1,6 diphosphate will get breakdown so as to produce glyceraldehyde 3 phosphate and dihydroxy acetone phosphate, and once again the dihydroxy acetone phosphate will convert back into glyceraldehyde 3 phosphate. So this is fourth and fifth reaction, but now we shall write the fourth reaction. So now in the fourth reaction, fructose 1,6 diphosphate will split to give two 3 carbon atom molecules. One is glyceraldehyde 3 phosphate and other one is dihydroxyacetone phosphate. And for that we shall write the reaction. So here in presence of the enzyme aldolase. So in presence of the enzyme aldolase, fructose 1,6 diphosphate will break down to give glyceraldehyde 3 phosphate HOH CH2OPO3 2 minus. This is glyceraldehyde 3 phosphate. And along with glyceraldehyde 3 phosphate, we will be getting dihydroxyacetone phosphate, which can be written in this way: C double bond O and CH2OH. So this is dihydroxyacetone phosphate. Simply we can write it as DHA. So this is the fourth reaction. So what we can say, in fourth reaction there occurs cleavage of fructose 1,6 diphosphate so as to get glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. So it is the process of cleavage of fructose 1,6 diphosphate. So the 1,6 diphosphate will undergo cleavage so as to produce two 3 carbon atom components. So this is fourth reaction. And in fifth reaction, that what we have given in the flow chart, in the fifth reaction, the dihydroxyacetone phosphate, which is produced in reaction number 4, will convert back reversibly to glyceraldehyde 3 phosphate. That will be the fifth reaction. And now we shall write the conversion of dihydroxyacetone phosphate into glyceraldehyde 3 phosphate. Okay, now the dihydroxyacetone phosphate, which is produced in the fourth reaction, will reversibly convert back into glyceraldehyde 3 phosphate, that is CHO, CHOH and CH2OPO3 2 minus. This is glyceraldehyde 3 phosphate. So here the dihydroxyacetone phosphate has been converted into glyceraldehyde 3 phosphate. Once again, these two are the isomeric forms. So because these two are isomeric form and both of them are triose sugars, both. In fifth reaction, the dihydroxyacetone phosphate, which is produced in reaction number 4 through the cleavage of fructose 1,6 diphosphate, will be reversibly isomerizes to produce glyceraldehyde 3 phosphate. But you can notice both are 3 carbon atom molecules and both are the triose sugars. These two are the derivatives of triose sugars, but both of them are in phosphate form and they are interconverting with each other. So in order to make the conversion of dihydroxyacetone phosphate into glyceraldehyde 3 phosphate, the enzyme that should be utilized will be, since I said these two are the triose sugars, derivatives of triose sugars, so they are triose, but since they are in the form of phosphate, so triose phosphate, and they are interconverting with each other. They are the isomeric forms to each other. So the enzyme will be triose phosphate isomerase is the enzyme name that converts dihydroxyacetone phosphate into glyceraldehyde 3 phosphate. So what happening here? Here they're occurring isomerization of dihydroxyacetone phosphate, that is DHAP, dihydroxyacetone phosphate. So now we have given the first 5 reactions of glycolysis, that what we simply call as preparatory phase. And you can notice, so in the first 5 reactions we have observed that there occurs utilization of 2 ATP molecules in reaction number 1 as well as in reaction number 3. So do remember there occurs generation of consumption of 2 ATP molecules. So here 1 ATP will combines to give 1 ADP molecule over here. So these are the first 5 reactions of EMP pathway of glycolysis. And now we shall see the production of pyruvate starting from glyceraldehyde 3-phosphate. And here you can notice, so from one glucose molecule, totally we are having 2 glyceraldehyde 3-phosphate molecules. So one glucose molecule will produce 2 glyceraldehyde 3-phosphate molecule, and now the reaction will proceed from glyceraldehyde 3-phosphate molecule, that is 2 glyceraldehyde 3-phosphate molecules. So now the sixth conversion, that is the glyceraldehyde 3-phosphate will first converts into 1,3 diphosphoglycerate. So here glyceraldehyde is converting into glycerate, means an aldehyde is converting into the form of ester. So if an aldehyde has to be converted into ester, there should occur oxidation of the aldehyde. That means we need an enzyme that performs dehydrogenation. So now we shall see how this glyceraldehyde 3-phosphate will converts into 1,3 diphosphoglycerate, and which is the enzyme material that actually takes place in that conversion. Now the glyceraldehyde 3-phosphate, which is produced by the glucose molecule, actually there are 2 glyceraldehyde 3-phosphate molecule, and this glyceraldehyde 3-phosphate molecule in presence of inorganic phosphate and reduced coenzyme, that is NADH, nicotinamide adenine dinucleotide hydrogen, NAD+. So in presence of inorganic phosphate and nicotinamide adenine dinucleotide, reversibly, actually for each glyceraldehyde 3-phosphate we need, now for each now we have total of 2 glyceraldehyde 3-phosphate molecules, and each glyceraldehyde 3-phosphate molecule will utilize 1 inorganic phosphate and 1 NAD+, that is nicotinamide adenine dinucleotide. So for 2 glyceraldehyde 3-phosphate molecule, there will be 2 inorganic phosphates and 2 NAD+. And now, as I said, the aldehyde group which is present in glyceraldehyde 3-phosphate should undergo oxidation, and oxidation of this glyceraldehyde 3-phosphate will convert this glyceraldehyde 3-phosphate into 1,3 diphosphoglycerate, CHOH, and this third position phosphorylation will remain as it is. So glyceraldehyde 3-phosphate will converts into 1,3 diphosphoglycerate. So here there occurs oxidation of aldehyde group into ester form. So phosphate group, 2 phosphate groups will be utilized, so that we will be getting 2 molecules of 1,3 diphosphoglycerate, and 2 NAD+ units will get changes into reduced coenzymes, it is 2 molecules of 2 NADH plus 2 H+. So this is the 6th reaction that occurs in case of EMP pathway of glycolysis. And now we shall see what is the 7th conversion. The 7th conversion is 1,3 diphosphoglycerate will get changes into 3-phosphoglycerate, means here we are removing 1 phosphate unit so as to get only 1 phosphate unit, and that would be present at the position of 3. And now we shall see how that actually happens. Before going to that, we shall see which is the enzyme that actually taken part in converting glyceraldehyde 3-phosphate into 1,3 diphospho-glycerate. As I told you, while giving the name of an enzyme, first we should know which is the substrate material that is taking part in the reaction and what kind of reaction is taking place. And here I said we are observing the oxidation of the aldehyde group. So oxidation will be performed by an enzyme that what we call as dehydrogenase. And the reactant, the substrate material is glyceraldehyde 3-phosphate. So the enzyme we use over here is glyceraldehyde 3-phosphate dehydrogenase. This is the enzyme we use over here, and what happens over here? Here occurs oxidation of glyceraldehyde 3-phosphate. Oxidation of glyceraldehyde 3-phosphate so as to produce 1,3 diphosphoglycerate. And now we shall see the seventh reaction, that is conversion of 1,3 diphosphoglycerate into 3-phosphoglycerate. Okay, the seventh reaction is conversion of 1,3 diphosphoglycerate into 3-phosphoglycerate. So we are having two molecules of 1,3 diphosphoglycerate, and you have noticed that in the first reaction and in third reaction there occurs utilization of two ATP molecules so as to produce two ADP molecules as a whole, and those two ADP molecules will comes into picture over here, and in presence of the enzyme called as phosphoglycerate kinase. It's a different case of kinase. So in case of presence of the enzyme kinase, we have noticed that phosphorylation, but this is a different kinase enzyme, a special case. There it causes removal of the phosphate unit from diphosphoglycerate from an ester. So we are having totally two 1,3 diphosphoglycerate which is present at first position and third position. Each ADP molecule will receive one phosphate unit from first position, and that converts into ATP, and this will change us into 3-phosphoglycerate that we shall write. So here the first position will remain in the form of ester, and the second position will carry H and OH, and the third position will remain with CH2O PO3 2 minus. As a whole we are having two molecules, and these two ADP molecule will get converts into 2 ATP. So here you can notice that this is the substrate molecule. We are having ADP, and from substrate phosphate unit is transferring into ADP so as to produce ATP. That means we can clearly say that this reaction is a best example for substrate level phosphorylation. So here 1,3 diphosphoglycerate is high energy phosphate, and that causes substrate level phosphorylation of ADP so as to produce ATP. This is the seventh reaction of glycolysis. And the eighth reaction is conversion of 3-phosphoglycerate into 2-phosphoglycerate. That means the position of the phosphate unit is getting deferred. That means there occurring some mutation within the molecule. So 3-phosphoglycerate will be converting into 2-phosphoglycerate. And now we shall write the reaction for it, and we shall see which is the enzyme that actually involves in making the conversion of 3-phosphoglycerate into 2-phosphoglycerate. Now the 3-phosphoglycerate molecule, 3-phosphoglycerate, which is produced in case of reaction number 7, now will undergo changes so as to produce 2-phosphoglycerate. Means from this third position this phosphate unit should go for second position at the part of OH. So first we shall write the complete conversion, that is COO minus. So C, or else let me write in this way, CH, O PO3 2 minus and CH2OH. So 3-phosphoglycerate has been converted into 2-phosphoglycerate. So the same thing, there will be no change in the molecular formula. The only thing is the position of the phosphate unit has been shifted from third position to second position. That means in a simple biological language we can say there occurs some mutation, some internal conversion, some internal changes. So phosphoglycerate has been undergone some kind of mutations so as to produce 2-phosphoglycerate. So the enzyme material should be phosphoglycerate mutase. This is the enzyme that actually causes conversion of 3-phosphoglycerate into 2-phosphoglycerate, and the reaction will actually occurs in presence of the activator Mg2+. So what happened over here? There occurs a shift of phosphate unit from third position to second position of the phosphoglycerate molecule. So this is simply shift of PO3 2 minus phosphate unit from C3 to C2 position. So this is the thing that occurs in case of reaction number 8. And now we shall see what is the reaction number 9. The reaction number 9 is the 2-phosphoglycerate which is produced will be changes into phosphoenol pyruvate. So there occurs production of an glycerate into enol form. So that we shall see in reaction, and similarly we shall see what is the enzyme material that actually causes conversion of 2-phosphoglycerate into phosphoenol pyruvate. Okay, in ninth reaction, this 2-phosphoglycerate reversibly changes into phosphoenol pyruvate in presence of the enzyme enolase under the influence of the activators Mg2+, or even in some cases it may be Mn2+. So in presence of the enzyme enolase, 2-phosphoglycerate will get changes into phosphoenol pyruvate minus. So this is the simplest structure of phosphoenol pyruvate. So this is the penultimate material in case of glycolysis. So 2-phosphoglycerate will get changes into phosphoenol pyruvate reversibly in presence of enolase. And here you can notice there occurs dehydration of phosphoglycerate in presence of the enzyme enolase, and that causes a distribution of the energy within 2-phosphoglycerate molecule, and that results in the formation of phosphoenol pyruvate, which can be simply written as PEP, phosphoenol pyruvate, which contains a high energy enolic phosphate. This is the 9th reaction. And the next, that means the 10th reaction in glycolysis is conversion of phosphoenol pyruvate irreversibly to produce pyruvate. And that we shall see how the conversion of phosphoenol pyruvate to pyruvate will happen. Here we get 2 molecules of 2-phosphoglycerate will produce 2 molecules of phosphoenol pyruvate, and 2 molecules of phosphoenol pyruvate will consume 2 molecules of water and 2 molecules of ADP so as to convert into the form of CH3 C double bond O COO minus, that what we call as pyruvate. And here in presence of water, ADP will receive the phosphate unit, and that results in the conversion of 2 molecules of ATP. Okay, so this is the 10th reaction, and the enzyme that take part in making the conversion of phosphoenol pyruvate into pyruvate is pyruvate kinase. in presence of the enzyme pyruvate kinase, there occurs conversion of phosphoenol pyruvate into pyruvate. Here also you can notice this is the substrate molecule. We are rich in phosphate, and that phosphate will be transferred into ADP so as to produce ATP. That is, once again here we are observing substrate level phosphorylation of phosphoenol pyruvate to produce pyruvate molecule. So till now we have given totally 10 reactions, and you can notice that in the first 5 reactions we have noticed the consumption of ATP molecule, and in the next 5 reactions we have noticed the formation of ATP and even formation of the reduced coenzyme, that is NADH. We know that 1 unit of NADH is equals to 3 molecules of ATP. So till now, whatever so far reactions we have given, these are the reactions that actually occurs in case of aerobic glycolysis. And now we shall see what will happen to the pyruvate molecule when the reaction occurs, when the glycolysis occurs in anaerobic condition. We said in case of anaerobic condition, the pyruvate molecule which is produced in the continuous stages of glycolysis will undergo reduction so as to produce lactate. In anaerobic condition, we said that the pyruvate molecule which is produced in the glycolytic pathway will undergo reduction so as to produce lactate. And now we shall see how that reaction will actually occur and which is the enzyme that actually take part in the conversion of, in a strictly speaking way, the reduction of pyruvate into lactate molecule. So now we shall see the reaction of conversion of pyruvate into lactate. So first, pyruvate, which is having the structural formula CH3 C double bond O COO minus. This is the pyruvate molecule, and this pyruvate molecule in anaerobic condition reversibly get changes into lactate, that is CH3 OH H and COO minus. This is the simplest form of writing the structure lactate. So here you can notice the conversion of pyruvate molecule into lactate. So totally 2 molecules of pyruvate will be produced, and that will be produced as 2 molecules of lactate. So, or else we can say in simply 1 molecule of pyruvate will produce 1 molecule of lactate. So here you can say in case of conversion of pyruvate into lactate, there occurs consumption of reduced coenzyme with proton so as to produce NAD plus, and that causes conversion of pyruvate into lactate. And as I said, the reaction is reversible in nature. The same NAD plus will causes dehydrogenation from lactate so as to produce NADH and H plus, and that causes once again conversion of lactate into pyruvate. So as a whole, what we can say, under anaerobic condition, the pyruvate is reduced to lactate, and the reaction that causes interconversion of pyruvate into lactate is lactate dehydrogenase. This is the enzyme that actually causes conversion of pyruvate into lactate. And remember, the NAD plus which is regenerated will allows glycolysis to continue in the absence of the oxygen. This is the reduction of pyruvate into lactate. So thereby we have given details of reaction pathways in both aerobic condition as well as in case of anaerobic condition. And now it's the time to calculate the energy yield of glycolysis in both aerobic as well as in anaerobic condition. That's what we used to say as energetics of the glycolysis. So now we shall calculate what is the total amount of ATP produced in both aerobic as well as in anaerobic glycolysis. Now we shall calculate the energy yield of glycolysis in both aerobic as well as in anaerobic condition. So firstly, we shall calculate the energy yield of glycolysis in aerobic condition. So in case of aerobic condition, the glycolytic pathway will stop at formation of pyruvate. So you can notice in the first step, in conversion of glucose into glucose 6-phosphate, there occurs consumption of ATP. And in the third reaction, in conversion of fructose 6-phosphate into fructose 1,6 diphosphate, there occurs consumption of 1 ATP molecule. But in sixth reaction, there occurs conversion of glyceraldehyde 3-phosphate into 1,3 diphosphoglycerate, and from each glucose molecule we will be getting 2 glyceraldehyde 3-phosphate molecule, and from 2 glyceraldehyde 3-phosphate molecule we will be getting 2 NADH. And in seventh reaction, in converting 1,3 diphosphoglycerate into 3-phosphoglycerate, we have observed the formation of 2 ATP molecule. And at the last, in converting phosphoenol pyruvate into pyruvate, we have noticed the formation of 2 ATP molecule in case of aerobic condition. So here 2 plus 2 plus 2 NADH. 1 NADH is equals to 3 ATP molecules. Okay, so there are 2 NADH molecules, so there are 6 ATP molecule, 6 plus 2 plus 2. So totally 10 ATP molecules are there. So 10 ATP molecules have been produced in payoff phase, but there are 2 ATP molecules that are consumed in preparatory phase. So as a whole, what we can say, so 2 plus 2 plus 2 into 3 minus 2. So 2 plus 2, 4, 3 2s are 6 plus 10, 10 minus 2, the total of 8 ATP molecules have been produced in case of aerobic glycolysis. So the reaction numbers we have already said. And in case of anaerobic glycolysis, if you come to the case of anaerobic glycolysis, so the reaction will continue from pyruvate onwards so as to produce the lactate. The same thing will continue, but the thing is, in case of anaerobic condition, we can notice that in reaction pathway, that is, here you can see, in case of reduction of pyruvate into lactate, there occurs utilization of NADH so as to produce lactate. So each glucose molecule will produce 2 pyruvate molecule, and each pyruvate molecule consumes 1 NADH in producing lactate. So in glycolytic pathway up to pyruvate, there were 2 NADH molecules have been produced. So 2 pyruvate molecules will consume 2 NADH. 2 pyruvate molecule will consume 2 NADH plus, and so as to produce 2 NAD plus in producing 2 lactate molecules. So the 2 NADH molecules which are produced in the glycolytic pathway will get utilized in converting pyruvate into lactate. So as a whole, we can say that the number of ATP molecules that could be produced in anaerobic condition will be 2 ATP molecules in converting 1,3 diphosphoglycerate into 3-phosphoglycerate plus 2 ATP molecules in converting phosphoenol pyruvate into pyruvate, and in the first and third reaction there occurs utilization of 2 ATP molecules. So 2 plus 2, 4, 4 minus 2. So in anaerobic condition, the total number of ATP molecules produced in glycolytic pathway is equals to 2. So the energy yield of glycolysis in aerobic condition is 8 ATP per glucose molecule and 2 ATP in anaerobic condition per glucose molecule. So these are the energy yield calculation of the glycolysis. So this is about carbohydrate metabolism, in particularly about glycolysis. So if the video is useful for you, please like it, share it, and do subscribe to the channel and press the bell icon to get the notification for any recent update in the channel. Thank you very much.